Hello friends, today we will discuss about the structure and functions of plasma membrane. This is the electron micrograph of plasma membrane. So what is the definition of plasma membrane? The plasma membrane of a cell is a network of lipids and proteins that forms the boundary between a cell's content and outside of the cell. It is also simply called the cell membrane. Its main function is to protect the cell from its outside environment. It is semi-permeable and regulate the materials that enter and exit the cell. So what are its function? The first main function, it creates boundary between the cell and outside environment. It localizes specific function. It helps in transport of molecule from outside to inside and vice versa. It helps in signal detection. It helps in cell-to-cell communication, and it helps in cell-to-cell adhesion. These are synthesized in the endoplasmic reticulum and Golgi apparatus. So there are various models which describe the structure of plasma membrane. These are lipid and lipid bilayer model, unit membrane model, fluid mosaic model, and Daniell model. According to lipid and lipid bilayer model, there are indirect information which explain the structure of plasma membrane. In 1902, Overton observed that substances of soluble in lipid could selectively pass through the membranes. On this basis, he stated that plasma membrane is composed of a thin layer of lipids. Subsequently, Gotter and Grandel in 1926 observed that extracted from erythrocyte membranes was twice the amount expected if a single layer was present throughout the surface area of these cells. On this basis, they stated that plasma membrane is made up of double layer of lipid molecules. But this model could not explain the proper structure of plasma membrane, but they have put the foundation for the future models. Second model is unit membrane model. This was proposed by Davison, Daniell, and Robertson. When surface tension was measured of plasma membrane, it suggests presence of proteins. It was suggested that surface tension of cells is much lower than what one would expect if only lipids were involved. It may also be observed that if protein is added to model lipid water system, surface tension is lowered. This suggested indirectly the presence of proteins. Only this basis, Davison and Daniell proposed that plasma membrane contained a lipid bilayer with proteins on both surfaces. Initially, they supposed that proteins existed as covalently bonded globular structures bound to the polar ends of lipids. Subsequently, they developed the model in which the proteins appear to be smeared over the hydrophilic ends of lipid bilayer. This model makes its popularity for a long time. The definite plasma membrane of 6 nanometer to 10 nanometer thickness was observed on surface of all cells, and plasma membrane of two adjacent cells were found to be separated by a space of 1 to 15 nanometer wide. It was also observed that the plasma membrane of most of the cells appear to be three-layered. Two outer dense layers, which are 2 nanometer thick, and middle layer is 3.5 nanometer thick. The early ideas of Gotter and Grandel and those of Davison and Daniell were first formalized by Robertson in 1959 in form of his unit membrane concept. So this concept of unit membrane with three layers, two proteins and one lipid only supported the concept proposed earlier by Davison and Daniell. Third model is fluid mosaic model. In 1972, Singer and Nicholson have given the fluid mosaic model. According to this model, there are peripheral proteins which are like the mosaic, like the tiles, which are embedded in the phospholipid medium, which is fluid, which is a fluid medium. So these proteins are tiles which are predicted to be mobile in phospholipid C. That's why it is called fluid mosaic model. The fourth model is Daniell model. According to this model, lipid and intrinsic proteins are present in a mosaic arrangement, and Biological membranes are semi-fluid so that lipids as well as intrinsic proteins are able to make movements within the bilayer. This concept of fluidity implies that lipids, proteins, and oligosaccharides are held in their positions by means of non-covalent interactions. On this basis, it can be stated that components can be dispersed by solvents or detergent without breaking any bond. So here is fluid mosaic model. The fluid mosaic model consists of three components: phospholipids, proteins, and carbohydrates. The phospholipids are main fabric of the plasma membrane, which are amphipathic or dual-loving. So these phospholipid molecules have hydrophilic heads, which are water-loving areas, which are on the aqueous or watery side, that is outside of the cell. And the hydrophobic or water-hating molecules tend to be non-polar; they are inside of the cell. A phospholipid molecule consists of a 3-carbon glycerol backbone with 2 fatty acid molecules attached to carbon 1 and 2 and a phosphate containing group attached to the third carbon. This arrangement gives the overall molecule an area described as its head, and the two fatty acid chain form the tail of phospholipid molecules. Here you can see the structure of phospholipid molecule with glycerol its backbone, which has attached phosphate group and 2 fatty acid. These phospholipid forms the micelle-like structure in detergents and solvents. The membranes uh, are having the property of fluidity. The phospholipid can move laterally within the bilayer or from one layer to another layer. The proteins are of two types: intrinsic proteins and extrinsic proteins. The extrinsic protein are present on the outside surface of the plasma membrane, and intrinsic are present embedded within the phospholipid bilayer or inside of the cell. These can be integral monotopic proteins, single-pass protein, multi-pass proteins, multi-subunit protein, peripheral membrane protein, and fatty acid or prenyl anchor. These proteins can act as channels and pumps which helps in transport of molecules. They act as links to the structure proteins. They act as receptors. They act as enzymes and helps in energy generation. The third component is carbohydrate. Carbohydrate, they are always found on the exterior surface of the cells. There can be 2 to 60 monosaccharide units which can be either branched or straight. The peripheral proteins can be joined with the carbohydrate. They can be glycoprotein, and if it is connected to lipid, they form the glycolipids. Both glycoprotein and glycolipids form the glycocalyx, which is very important for the cell recognition in immune system. The common carbohydrates are beta-D-galactose, beta-D-mannose, N-acetyl beta-D-glucosamine, and sialic acid. So here I have attached one video for the simplicity structure of plasma membrane. So this is showing the phospholipid bilayer. With its head and tails. So this membrane forms the channel for the different molecules to pass from it. It has various integral proteins which act as channels and pumps.